So chapter 3 is the cell, and the cell is a fundamental unit of life. Anything less than that, we cannot call it life. And we talked about that before. When you go less than that, like you have a mitochondria, it doesn't do anything. It's not life. It's not the life form. The least is the cells. You put cells together, become a tissue and organs and so on, but this is the smallest of all. This is if we're talking about humans, but bacteria, for example, is just one cell, and so on. So this is what we will be covering, which is basically everything about the cells with different types. Do you guys know anything about the cells? What do you know about the cells? Like any component in the cell?
There's a nucleus.
Nucleus inside.
Very tiny.
Huh?
Small.
Small. Okay, but what else? The components. Cell wall. Cell wall in certain things, but cell membrane is in everything. There is a difference. So nucleus and cell membrane. What else? Ribosomes. That's 3. Mitochondria. You know the mitochondria? So that's 4. Golgi apparatus. Okay, we will cover all of this. But generally speaking, any living organisms consists of cells from 1 to trillions. Sometimes it's only 1 cell. Can be more, all the way up to billions or trillions. But the cells are the fundamental unit of life. This is the least. This is the smallest. Anything less than that, like organelles, this does not count. It's part of the cells. So the cells are the fundamental units of life. There are 2 types of cells that we're going to learn. And the first type is called prokaryote, and the second one is called eukaryote. If you look at it from the medical terminology that I usually focus on, because it will make things make sense, make more sense when you understand it. What's pro and what's karyote, and what's eu, which is EU, and what's karyote again. Pro means it's a prefix that means before or earlier to, before. EU is a prefix that means normal. Karyote, the exact translation is colored, but they refer to the nucleus. So when you say karyote, you're talking about the nucleus. So what does that mean? This is not here, I'm just explaining it to you. Prokaryote means this is too old. It was the first thing that's evolved in the very beginning before it advanced enough to have a nucleus. So prokaryote means before the development of the nucleus. It does not have a nucleus. The bottom line. This is not the only difference. I'm just telling you where the name came from. Is that clear? Eukaryote means normal nucleus. So anything that have a nucleus, we call it eukaryotes. There are other things that will go without saying, but this is the bottom line, or where the name came from. Prokaryote, before the karyote, before the nucleus. You do not have a nucleus. We call you prokaryote. Later on, with evolution, things started to evolve from that, and you started to make a nucleus. Now we call it eukaryote. Is that clear so far? This is just the start. This is the huge point, the big point. Beside that, usually the prokaryotic cells are small, simple cells, and it's usually 1. Prokaryote is usually 1 cell. Okay? Does not have an organelle. And it started 3.5 billion years. This is like the ancient one. This is the oldest one. This is the very primitive ones, from which evolution happened, and you get more advanced, which is the ones that have the actual nucleus. Okay? If you compare that to the eukaryotes, it's larger, it's more complex, it have organelles that most of it is covered by a membrane. This does not have organelles, does not have membranous organelles. They have membranous organelles. This is 2.1 billion, so obviously this is more advanced. This came later, and it can be unicellular and it can be multicellular. So which one is unicellular only?
Pro.
Eukaryote can be Both can be unicellular, can be most of the time it's multi, but it can be either. So what do you think about yourself? Are you a prokaryote or eukaryote? Eukaryote, of course. You're a very advanced eukaryote, right? But if you look at this, this is a prokaryote, and the example here is bacteria. Bacteria is a prokaryote. So if you look at it here, in the middle here, you have something. You think it's a nucleus. It is not, because when we say nucleus, which is eukaryote, means the classic nucleus. What's the classic nucleus? By definition, it means it has the nucleoid material or chromosomes surrounded by a membrane. This is the only time when I say nucleus. Okay? So what if I have chromosomes inside a cell? It is not a nucleus. It has to be surrounded with a membrane. Is that clear enough? So anything will have chromosomes anyway. Anything will have chromosomal material, or it have a nucleoid material. Okay? It will have it. It will have chromosomes anyway. But is it surrounded by a membrane or not? If it's surrounded by a membrane, this is the classic definition of a nucleus. Other than that, you will have some chromosomes, but look at this. There is no membrane around this. So we call it nucleoid. What's nucleoid? Nucleoid. Nucle is nucleus. Oid means similar to. It's a suffix. I don't know if you did medical terminology or not, but I'm giving you some prefixes and suffixes as we go. The more you collect, the easier it gets. If you're going to more advanced classes, you will see what I'm talking about right now. Just collect as much as possible of these prefixes and suffixes. So the suffix here is oid. So you can put oid after anything and it will mean similar to something. Similar to. So nucleoid means similar to the nucleus. Is it a nucleus?
No.
No, it is not. What kind of similarity? Chromosomes. Okay, it has some chromosomes, but not membranous. Other than that, you will have a membrane. If you don't have a membrane, you're not cell. Okay? If you don't have a membrane, if this does not have a membrane, it's not a cell. All cells, with no exception, on Earth, whether it's a prokaryote or eukaryote, as long as you call it a cell, you're talking about two things: a membrane from outside and chromosomes inside. If these chromosomes have a membrane, we call it eukaryote, which is a nucleus. If it does not, it's a prokaryote. Is that clear so far? Okay, so this is an example of the prokaryote, which is the bacteria, and this is an example of eukaryote. Eukaryote can be plants or animals. We can be classified under eukaryotic, which is animals, but this is an example of a plant cell. How to know this is a plant cell? I will explain it. But the most important thing that I want you to notice here is this is a membrane, and this is the nucleus. You see how there is a cover? You see this cover right here? This is a membrane. So if this is a nucleus or not? It has the membrane. It is the nucleus. That's why this is a plant cell and it is eukaryote. How did you know it's eukaryote? There is a membrane. And inside, of course, there are chromosomes anyway. It goes without saying. Any cell on Earth will have a membrane, will have chromosomes inside. Okay? Anything. Whether the chromosomes are surrounded by a membrane or not, this is another issue. But you cannot even call it a cell if it does not follow this. Okay? But other than that, you will see other organelles here. You can see Golgi Apparatus, you can see the chromoplast, you will see other things. But I'm just talking about the basics now, and then we will move on. Okay? So did you get this so far? Are we following? Okay. So here is a prokaryote cell, and this category include bacteria and archaea, and you need to remember this. So what are the prokaryotes that you know? Bacteria and archaea. Bacteria and archaea. Okay? Does the bacteria and archaea have a nuclear membrane? No. Does it have chromosomes? Chromosomal material? It does. Does it have a membrane? A membrane, not a cell membrane. No, not a nuclear membrane. Cell membrane. Does it have a cell membrane? You don't call it a cell if it doesn't have a membrane. Okay? Anything you can add, plus or minus, but the bottom line is you have to have chromosomes, you have to have a membrane. cellular membrane. Okay? If you have chromosomal material without a membrane, I call it prokaryote. If you have it within a membrane, I call it eukaryote. Beside other things, but this is the basic one. So here are some examples of bacteria and archaea. And here is a bacteria, Spiralium volutans. This is something that if you get a sample of water, fresh water, and look under the microscope, you can see it. It's a bacteria. The other one is an archaea. So just remember that prokaryotes are either bacteria or archaea. The domain is prokaryotes, but you have both of those, bacteria and archaea. Eukaryotes can be plants, animals, plus fungi and protists. You need to remember this. You need to remember this. Plants, animals, this is what we all know, and add to that fungus and protist. So remember, is bacteria prokaryote or eukaryote? Prokaryote. How about archaea? Pro. How about fungus? Eukaryote. Eukaryote. Protist. Eukaryote. The protist is the only eukaryote that's one cell. Okay? And this is a human cell. This is an onion. I don't know if you guys are doing that in the lab or not. We used to do it in general biology. You can find like pieces of onion, and you have a slice, and you put it under the microscope, and this is what you're going to see. It will look like this. It have a cellular membrane. It have a nucleus with a nuclear membrane around it, and you see other things like chromoplasts and other things. So we already said that the prokaryotes are simple, small, single. Are the eukaryotes single? It can be single, it can be multiple, right? But prokaryotes are single. It's unicellular. And we said there is, in this domain of prokaryotes, have bacteria and archaea. Remember this? This is called the domain. Prokaryote is a domain. Eukaryote is another domain. Prokaryote is a domain that includes bacteria and archaea. Eukaryote is another domain that contain plants, animals, protists, and fungi. Okay? The prokaryotes are very old, like 3.5 billion years old. So this is obviously the oldest. And here is a bacteria that we looked at something similar to that before. The basics are you have to have chromosomal material, which is the nucleoid, and you have to have what again? Membrane, cell membrane, cell membrane. These are the basics. What else? We can talk about this. But if you don't have those two, it's not a cell in the first place. Clear? Okay, so we have nucleoid here, which is kind of similar to the nucleus that we have in humans and animals and others, but without a membrane. Okay? But does it have DNA? Yes, it does. It does have DNA, which is the chromosomes. We have the chromosomes. What else do we have in the bacteria? The inside of the membrane between the nucleoid, which is the DNA, and the membrane for outside. What's filling this gap? The membrane is outside and the nucleoid is inside. What's filling the gap? It's called the cytoplasm. Same thing in the human cells. In between you have a cytoplasm. Okay? What else can you have here? Here is something that they have, but we do not have it. They can have, beside the plasma membrane. What's a plasma membrane? It's a cell membrane. It's the same thing. Plasma membrane or cell membrane is the same thing. Cell membrane because it's the membrane of the cell. Plasma membrane because it's the membrane around the cytoplasm. That's why they call it plasma cells. Plasma cells. Are we following? Okay, so what else do we have if this is a prokaryote-like bacteria? Beside the membrane that you already have, you can have another wall, and you cannot confuse the cell wall with the cell membrane. The cell membrane is basic. You have to have it in every single cell, cellular membrane. But the cell wall is here, and in plants as well. Beside that, you might have a capsule outside. Did you get the idea? Do you have a membrane anyway? Anything have a membrane. Is that okay? If this is a prokaryote, can you have additional things outside of that? Yes. You can wear a jacket and another jacket, maybe. Wall and maybe a capsule outermost. The capsule is like the jacket outside. Okay? How about eukaryotes? It depends. But like animals and humans, you have the membrane only. Plants will have a wall, none will have a capsule. Is that okay so far? So we're talking about this now. You have a membrane. This is bacteria, prokaryote. Membrane. What's outside of the membrane? Cell wall. All of them have it. What is a plus or minus outside of the wall? Capsule. Maybe yes, maybe no. It depends. What else do we have here? We can have a flagellum like this. Flagellum means a tail to help them to move. And we can have pilus or pili like these. And these pili are something that help you to stick something. It goes like this, and you can stick it like this. So they can, like bacteria, come together, or the bacteria can stick itself on the wall of one of our cells or animal cells or anything. This is how they stay. Like if you think about it, this is a bacteria and it's going like this. It will come to your cells. Why does it stay? Why it doesn't like wash away? They stick themselves using this pilus. This is one of the uses. The other use is you can use it as a communication like this. If this is a bacteria and you have this, maybe things can leave out. Like if two of those, this is pilus from one bacteria and this is pilus from other bacteria, they can stick themselves together like this. So they stay together, right? But they can also exchange small things in between themselves. What is that small thing that we have? It's called the plasmid. The plasmid is just a circular, tiny DNA. Just not this. This is the main one. We can have little pieces, little circular pieces that are called the plasmids. Okay? So, and that explains, if you think about it, sometimes we hear like the bacteria is becoming mutants. We used to kill the bacteria, but it's not effective anymore. Drug resistance. Did we hear about this? Drug resistance. Where does it come from? There are different things that can come from, but this is one of them. Maybe a plasmid from this bacteria moved and go to the next one, so now it's not the same. So when you give the antibiotic, it does not find the regular structure. Okay? So these are the plasmids that can move, and these are little pieces, so it can travel, okay, through the pilus from one bacteria to the other. Okay? There are other uses for that. They use it in DNA or chromosomal engineering and stuff like that, but they use these plasmid. They can take the plasmid from the bacteria, they insert something in it, and they transfer it from person to person. There are other uses for it, but what I want you to remember here are the basics. So what is the plasmid? Plasmid is a small circular DNA, a small circular chromosomal material. Clear? It's have nothing to do with this big nucleoid material here. Are we following so far? So if you think about the prokaryotes again, some prokaryotes like bacteria, they live anywhere, right? You can see bacteria anywhere. You can see it in the water, you can see it in your own body. Whether it is normal or it's an infection, it can see it in animals, in plants, in anything, right? Bacteria can be found anywhere. But more importantly, in humans, you can have two types of bacteria. Are we following? Are we still following? Okay, you can see different types of bacteria. There are some bacteria that coexist with us, and these are non-harmful bacteria. Okay, sometimes it's even beneficial bacteria. Like in your colon, it's all over the place. It's everywhere.
Normal flora?
The normal flora, yes, normal flora. The normal flora is the bacteria that's not harmful. It actually can be beneficial. The best example of that is in our colon, we have some type of beneficial bacteria that makes vitamin K for us. To the extent of, believe it or not, if this is a patient who's admitted in the hospital and taking a big dose of antibiotics, killing the bacteria, you can have bleeding issue. Like you have a minor injury and you bleed, it doesn't stop. to that extent. This is our main source of vitamin K, and we call those the normal flora or the non-harmful beneficial bacteria. Other types, like you get sick, you have pneumonia, for example, from bacteria from outside. This is another type. But I was talking more about the normal commensal or non-harmful or beneficial or normal flora. Okay, so we already talked about the eukaryotes before, and we said the eukaryotes are bigger, more complex. They have a traditional nucleus that have a membrane, right? And you have a cell membrane from outside. This is the classic structure of any eukaryotic cell, beside other details that we will talk about, but it's more complex than the prokaryotes. And it has membranous organelles. Membranous organelles. It was in the first slide when we compared prokaryotes to eukaryotes. Okay? Eukaryotes are going to be different, but what is not different is the plasma membrane I mentioned, the nucleus that I mentioned. You can add to that the mitochondria too. It just all of them have mitochondria in eukaryotes. But the first two, all of them have it. All of them have plasma membrane, nucleus, and you can add to that mitochondria as well. So whether you're talking about an animal cell, whether you're talking, including humans, when we say animal, it implies to humans as well. It's the same structure. Or you're comparing it to plants. Do we have nucleus here? In plants, you do have a nucleus. Does it have a nuclear membrane? Yes. Same thing here, right? Do you have a cell membrane from outside? You do have a cell membrane from outside. Do you have a cytoplasm in between the nucleus and the membrane from outside? You do have a cytoplasm. What's filling it then? Is it a cavity? It's not a cavity. Okay? So these are the basics. You have mitochondria. You do have mitochondria. Then there are differences that we will talk about, but it does not include any one of those three. So what else can we find besides the nucleus that have a membrane? We will also see the nucleus of the nucleus. The nucleus of the nucleus. We call that nucleolus. Do not confuse nucleus with nucleolus. Okay, there is an O different. Okay, nucleolus is that tiny structure inside the nucleus. Sometimes we call it nucleus of the nucleus. Okay, and this nucleolus that's inside the nucleus have certain functions that we will talk about. What else can we find? We can find the mitochondria. And the mitochondria, most important thing to remember about it is energy. Energy. The mitochondria is our powerhouse. Energy. Mitochondria, energy. There are other things. Just remember this one. Energy. Okay? What is the nucleus? It contains DNA inside, right? It's a chromosomal material. And what's the importance of that? The DNA is like the headquarters. This is the manager's office that is managing the whole entire cell, the nucleus. Because the nucleus contains the manager. Who is the manager? DNA. Where is the office of the manager? Nucleus. Is that clear? The manager, the DNA, is inside his office, which is the nucleus, managing everything. And we will discuss the details later. But for now, nucleus is the manager sitting in the office, which is the nucleus, inside of which you have the nucleolus. So we have the mitochondria, you have the cytoplasm, which is this plasm in between the nucleus from one side and the membrane from the other side. We also have lysosomes. What are the lysosomes? Translation again, medical terminology again. Lyso means to dissolve. Lyso is a prefix that means to dissolve. Some, S-O-M-E, it means body. I'm not explaining anything, I'm just translating. Lysosome. So when you see some again, remember that I already told you. This is a suffix that means, what so means? S-O-M-E.
Body.
Body. Lyso, dissolve. So this is a body that dissolve other things. Clear? What are you going to dissolve? Anything that you don't need. It's located there. This is like your trash can. Okay? If you don't need anything, throw it in the lysosomes. And from the name, the lysosomes is going to dissolve it. Isn't this is a lysosome is a dissolving body, right? Like a shredder. If you don't need this paper, send it to the shredder, and then you are going to dissolve it, break it down, right? So exactly, lysosomes is like the shredder. Okay? Ribosomes. Ribosomes are the factories of protein. I can say it this way. Factories of protein. This is where you make the proteins. This is your factory. Okay? And there are two types of these ribosomes. Some of them are on the surface of these, do you see this blue network? Some of them will be stuck on the surface, and we call that the fixed ribosomes because it is fixed on the membrane or the wall or the membrane of this network, and this network is called endoplasmic reticulum. And the other ones are just scattered in the cytoplasm, free in the cytoplasm. So ribosomes are, what are the ribosomes? What's the importance? Factory of protein. This is where we make the proteins. Some of them are free, some of them are stuck on the membrane of this network. What is this network? Endoplasmic reticulum. Let's take it piece by piece. This is what science says. Learn this. Cut the word into pieces and you'll get it. Endoplasmic reticulum. Endo means inside. Plasmic means the plasm, which is the cytoplasm. Reticular means a network. Understand how they put this name. They found a network in the cytoplasm. So what should we call it? What is this? I'm looking under the microscope. This is my first time to discover this. Let's say thousands of years ago. And I'm looking under the microscope. I found a network in the cytoplasm. What should I call it? Endoplasmic reticulum. Of course, this is Greek and Latin. It's not English, right? Endoplasmic reticulum. So endoplasmic reticulum can have the ribosomes stuck on the surface, and if that's the case, it will look rough. Other parts does not have ribosomes on the surface. We call it smooth, like this. If you look at this, this is smooth, right? I don't have anything here, smooth. But if I put something like this, it will look rough, right? So part of it that have the ribosomes stuck on the surface, I call that the rough endoplasmic reticulum. The other part that does not have these ribosomes, it looks smooth endoplasmic reticulum. We have two types of these networks. Are we following so far? Are we following? Any questions? Okay. What else do we have? We have vesicles. What are the vesicles? The vesicles are like bags. Like in your house, if you have extra chicken or meat or something, you can put it in a bag and store it, right? You can put it in the fridge or something, right? If you have extras, you don't leave it in the kitchen, you put it in a bag. Vesicle is your bag to store things. Like what? Anything that's extras. Okay, any nutritions, any water, you can leave the water in there, right? Don't you sometimes you put some water in the fridge, store it in case if you need it. If you have extra food, you put it in a bag in the fridge. This is the vesicles. Okay? The other thing is the cytoskeleton. Cyto means cell. What's the cytoplasm? The plasm of the cell. What's the plasm? The filling. Does everything have a meaning here? Cytoplasm. What's cyto? Cell, a prefix that means cell. Plasm, the filling. So what's the cytoplasm? The filling of the cell, right? How about the cytoskeleton? This one right here. Cyto means cell. Skeleton more. Yeah, you're close, but skeleton. Did you see the skeleton before? Did you see the bony skeleton, your skeleton? So this is the skeleton of the cell. Like you as human, you have your own skeleton, right? But if you look inside the nucleus, it has its own skeleton. Why do you have a skeleton? The same reason the cells have skeleton. Why do you have a skeleton? If you don't have a skeleton, you're going to collapse, right? Can you like stand up like this without bone, without skeleton? Of course not. So the cells themselves have its own skeleton to keep the shape as it is. Otherwise, the cells are going to collapse, right? This is for the cytoskeleton. And there are different types of things that we'll talk about, but this is the main part. Keep the structure and also help you to move, help the cells to move, okay? So we talked about the endoplasmic reticulum. How many times of endoplasmic reticulum do we have? 2. One of them is smooth, and the other one is? Which one have the ribosome on the surface? And what's the function of the ribosomes? Protein. It makes protein. Some of these cells will have this flagellum. What's a flagellum?
Like a tail.
Tail. Yes, it's a tail. Some cells have it. Okay? Comparing that to plant cells. When you see plant cells, it looks exactly like this. Okay? Compare it to the plant cells. How can I tell that this is a plant? I have a membrane. I have a nucleus with a membrane. I have endoplasmic reticulum, smooth and rough. I don't think I talked about the Golgi apparatus. The Golgi apparatus is this part right here. I didn't talk about it. This Golgi apparatus. What's the Golgi apparatus? It's another organelle. I'm covering the organelles now. What kind of organelles did I talk about? I talked about nucleus, one of the organelles. Nucleolus. I talked about the mitochondria. This is one of the organelles, membranous organelles. Okay? I talked about the vesicles. This is an organelle, right? Lysosomes, vesicles, membrane, cytoskeleton. Each one of those is an organelle. Are we following? The last organelle is in the plasmic the Golgi apparatus. What's the Golgi apparatus? It is an apparatus for packaging and exporting. Imagine this is a real factory. Like, what's the function of the ribosomes? Produce protein. This is a factory of protein, right? If you go to any factory, let's say this is a factory that's making TVs, for example. Do you take it from the factory directly and export it? Or do you have to take it and finish it and color it and package it, right? Put it in a nice box, right? You have to do the finishing part. If you wanted to export it or if you want to send it somewhere, you don't take it from the machine and take it out, right? You can't do that. You have to do the finishing part. This is in any factory, right? Are you done with the TV? Yes, give it to me. It's missing part. I will add it. The color here is not perfect. Let me paint it. I'm going to put it in the box, and then I'm going to wrap the box. Ready, you can take it out, right? This is the function of the Golgi apparatus. So we call it the finishing and exporting deck, the finishing and exporting machine, okay, or the finishing organelle. So it will take the protein. So ribosomes are making proteins, right? The ribosomes will make the protein, and this protein is going to go on the plasmic reticulum until it reaches the Golgi apparatus. Golgi apparatus will take it. Will say, okay, I got the protein, but this is not ready to use. It's just coming to me from the machine, right? It's not usable yet. Let me take that protein, take a look at it. If there's any extras, I remove it, and then I'm going to package it, put it in a nice package, right? Wrap it. Now it's ready to use. This is the Golgi apparatus. Almost the same that I mentioned is in the plant cells, and here is the difference. You have 2 main differences. 3 actually. Differences. First difference. So if you look at it, you need to tell what it is. Okay? If you see a picture or something, you immediately tell me, is it a plant or animal? Which animal? Of course you don't know. But at least you can say this is a eukaryote anyway, and you can say this is a plant, this is an animal. Okay? What's the difference? Here is number 1. You have this chromoplast. The chromoplast is the green part of the plants. Isn't all the leaves green? At least during the not during the winter time or the fall, when it we start to lose it, right? Start to fall. But during the regular time, isn't it green? The green is the color of the chromoplast. What's a chromoplast? The chromoplast is responsible for photosynthesis. Do we know what's photosynthesis? Photosynthesis. Photosynthesis is simply the plants are using energy from the sunlight in order to make nutrients. Plants using energy from sunlight to make nutrients. And this is how they make the nutrients. Don't you, at least the minimal, if you have a plant, don't you water it at least? You water it, right? Otherwise it will die. So the plants will take the water. What else? Do we have carbon dioxide in the air already? Okay, so they will take carbon dioxide from the air, they will take water from the roots that you give them, they will put it together, and they will take some energy from the sun, and it's going to convert those two into nutrients. Okay? Polysaccharides specifically. Of course they do that for themselves, not for you, not for us. They make it for themselves. So they get the carbon dioxide, and actually the carbon dioxide is what we're breathing out, right? We take oxygen and we breathe out carbon dioxide. So the air is loaded with carbon dioxide, but the plants will take it and put water and use energy and convert this carbon dioxide and water into nutrients, okay? Mainly carbohydrates, right? So if you're eating apple, the apple obviously contains carbohydrates. If you ask yourself, how did the apple make that carbohydrates? It make it from carbon dioxide, water, and energy from the sun. And the reason why it does that, it make carbohydrates for themselves to feed themselves. This is their storage. But of course we cut it and eat it, right? But they produced it for themselves, okay? And this is how they do it, and this is how the photosynthesis occur. You have to have a chromoplast. Do you have chromoplast? Humans, do you have chromoplast? Of course not. So you cannot do photosynthesis. Plants can make photosynthesis for us, and that's why in, I think it was chapter 1 or something, when I told you the plants are the origin of everything. Because how can the animals, does the animals have chromoplast? Can they use the sun or something? Of course not. They cannot. So the plants eat the animals that already have, that already did the photosynthesis. And we either eat animals that already have, or we eat plants that already did the photosynthesis. So this is number one. Number two, you see this vacuole here? This big, huge vacuole? This is called the central vacuole. When you see the vacuole, or you see the chromoplast, this is a plant cell. The central vacuole is a vacuole. Vacuole means space or cavity. This central vacuole is, what is this? There is storage of extra water and nutrients. They put it in the form of a vacuole like this. Do you remember the vesicles that I talked about in our cells? When you store the extras, the bags, instead of having these bags, they have a big vacuole like this, and any extras, they put it there. Extra of what? Water, nutrients, whatever they have. Any extra, they put it in that vacuole. Okay? The last one here is the cell wall. Do we have a cell wall? Humans have cell wall? Do you have a cell membrane? Yes, but you don't have a cell wall. So this is the only eukaryote that have a cell wall beside the membrane, outside of the membrane. Can you have a capsule here? In plants? No, this is only in prokaryotes, in bacteria. Eukaryotes, all of them have a membrane. Plants have a wall plus the membrane. That's it. That's it. You cannot have anything beyond that. Capsule is in some bacteria. Okay? So animals have lysosomes. What does the plants have instead of the lysosomes? Animals will have lysosomes, they have vesicles. The plants will have chromoplast, they have the cell wall, they have the central vacuole. Can you tell this is animal or not? Three things. You have the chromoplast, you have the cell wall. I'm not talking about the membrane. The wall is outside of the membrane. Okay? The number three is the central vacuole. As far as I remember, you're supposed to do that in a lab, like onion or something, and you look, and you should see all of this. If you look at the cell itself, you will see the following. I already gave you some information about the different components of the cell, but we have a little bit of details, not much. We already covered most of it. So let's go one by one. Do you know that all cells are surrounded by a membrane? Whether it's a prokaryote or eukaryote, right? All of them have a membrane. This membrane can be either called cell membrane or plasma membrane. Why do you have a membrane? The membrane that's outside, surrounding the cells, surrounding the cytoplasm, to regulate what goes in, what goes out. Don't we have a wall to this classroom? Right? Do you have a door? If this door is closed, can you get in? No. So that's why you have a wall. So this is how we determine who is allowed to get in and who is allowed to get out. Right? This is the membrane. So the membrane is surrounding the cells to regulate what can goes in and what can go out. This is why we have a membrane, controlling the passage of anything. And we need to go a little bit in details in the structure of the cell membrane. Plasma membrane or cell membrane is the same thing, isn't it? Isn't the cell membrane and plasma membrane the same thing? Yes. So plasma membrane or cell membrane, what does it consist of? It consists of a double layer of phospholipids. And I remember we talked about the phospholipid before. Are you guys studying as we go? Do you remember the phospholipid? The head is? It is hydrophilic, and it is? Is the lipid hydrophilic? The head is phosphate. It's called phospholipid. What does it mean? Phosphate and lipid. The head is phosphate. The tail is lipid. And I even did that, remember? The head and the tail. The head and the tail, and the two layers are like this. You see this? Head, tail. So where is the head? Outside and inside, right? So let me draw this. Head, tail, head, tail, head, tail, head, tail. Okay, it is one layer. This is the second layer. And so on. So you can go like this, a complete circle. You see the two layers? Double layer of what? Phospholipid. What's the head? Phosphate. Is that hydrophilic or hydrophobic? Philic. Anything that's not lipid is philic. And the head is phobic. Is it lipid? Lipid and phobic. Did you notice that the tails are touching each other? Like coming toward each other? There is a reason for that. It's coming closer to each other, but this is not the reason. The reason is I want the heads to be outside and inside. Why? Because the inside is the cytoplasm, isn't it? Cytoplasm is mainly water. Mainly water. So which side can interact with the water? The hydrophilic or the hydrophobic? Which one can interact with the water? Philic or phobic? Philic. Yes. So it's the head, right? The head. Here is another cell. And the same thing here. Okay? And in between the cells, you have another fluid. This fluid is called extracellular fluid. What's extra means? Extra. Outside. It's in between the cells, but we call it extracellular, means outside of the cells. This fluid here is also watery. It's water, mainly water, and other things. So which head can interact with this? Which part? The hydrophilic part. Now I have the inside is all lipid, right? This is also for a reason. Did you understand now why I'm putting this in front of each other like this? Why I'm putting the tail toward the tail? And the heads are outside and inside. The heads are hydrophilic, interacting with water inside and water outside. Make sense? Yes. The lipid is inside. I put it there to act as a barrier. So nothing can go in through it except if it is lipid soluble. There are other ways for things to get in and out, but simply you have a double, two layers here, the tail and another tail. You have those tails inside. So if something is trying to go like this, let's say this is water, okay? Look at this. This is water. It will go to the head. It can interact with the head because the head is hydrophilic, right? But can it cross? No. You cannot cross in a lipid. Can the water cross in the lipid? Can the lipid cross in the water? Of course not, right? So things will go like this, interact with the head, and stop. Okay? You stopped it. Then what? Then it depends. If you need it, you will allow it to get in some way, not the regular way. Okay? Did you get this? You are allowed to get somehow, but it doesn't go like this automatically. You have to open for it, and I will talk about this. Look at this section of the membrane. What is this? The phosphate, which is the head. And what is this? The lipid, which is the tail. Same thing here, right? Do you see how this whole inside is full of lipid? Nothing can go through it, except if it is lipid soluble, something that's lipid soluble, or you allow it somehow, and I will talk about this. Beside that, the other part of the membrane is you have some proteins. So the main structure is 2 layers of phospholipids plus proteins in different spots. So this is one of the proteins. Why do you have a protein like in the middle of those 2 layers like this? Because it acts like a door. Okay? If this was like this classroom and you don't have a door, you cannot communicate, right? So we have these doors, we call it the channels. Okay? So if you wanted something, and that something was hydrophilic, if that something is hydrophilic, and you cannot get it in, like water, can you get water in the cells?
No.
Can it cross the lipids? No. But do you need water inside the cell? Of course I do. So that's why we have these proteins, like you have exceptions. Your water you cannot get in, but I need you. So I will get you in from this type of proteins, transport proteins. Okay? Beside that, so we said it's double layer of phospholipid, and we said proteins, right? What else? We can have some carbohydrates and some cholesterol as well. One more time, the membrane is 2 layers of phospholipid. This is the main part. What else do we have? Protein, carbohydrates, and some cholesterol. Okay? Why do you have protein extending out, carbohydrate extending out like this? It can act as a receptor outside, like you put camera outside so you see what's happening outside. If somebody's coming, you can buzz, you can knock on the door from outside. This is one way. The other way is it makes the cells can stick, or things can stick with the cells. Okay? Now, if you're transporting something through the membrane, okay, we will have some more details to talk about. If that something is lipid soluble and small enough, it will go. No help needed. It will just go. Like, can I open this door? I have this, so I'm lipid soluble, small. I can do like this and open the door. Before I come, can you enter to this classroom? You cannot. So I have to open for you, right? So if this was lipid soluble and small enough, it will just pass. Okay? What if it is not? Can you transport? Can you allow it to get in? There are ways. Like I open the door for you. There is another way. So look at this. You will see. This is just an example. If you have a club, and think about it this way. We are going to club. Okay? If this club, or you can say it's a classroom, whatever it is. If this is a club, right? And I go to the club and the club is full of members. Is it easy for you to get in?
No.
No. Is it easy for them to go out?
Yes.
Yes. We call that concentration gradient. Concentration gradient. It moves from more to less. Make sense? Just open the door if it is full. Once you open the door, somebody will get out, right? If you try to get in, that's really hard because you're moving against concentration gradient. Did you understand my idea? If you try to push yourself in, it is full. So this is hard because you're moving across concentration gradients. Okay? So again, the membrane is there outside to regulate the entrance and the exits of anything. Okay? So there are things that can go through it, through the membrane, if it is small and lipid soluble. It can pass through. Okay? So this is green. Other things are yield. Like, I'm not saying no, but just yield here and let me see if you can go or not. And other things are you are not allowed to get in. Okay? So, and we call that selectively permeable. What's selectively and what's permeable? Selectively means you select what is allowed and what's not. Isn't it? Select. Permeable means able at the end, means ability. So permeability, the ability to permeate, to cross. Selective permeability means who is selectively allowed to get in and who is not. So that's the characteristic of this cell membrane. It's selectively permeable. Is it impermeable? You know what impermeable means?
You can't.
You cannot. Yes. I told you before that prefixes and suffixes are going to help you. Whether writing it or not, it's up to you. I'm just telling you what is going to help you later on. When you put IM, AN, or IN, or A at the beginning of any words in Greek or Latin, it means no. Okay? Impermeable, unpermeable, impermeable, this is not true. I'm just giving you examples. I can give you other examples. But if you put any one of those as a prefix before anything, it means no. So what impermeable means? You cannot cross. Is that a characteristic of the membrane? It's impermeable, like nothing can go through it? No, that's not true. Is it freely permeable? Anything can go through?
No.
Not that either. It's selective permeable, in between. Certain things are allowed, certain things are not allowed, and this is the most important characteristic of the membrane, selectively permeable. You allow things and you do not allow things. So let's say you are trying to get something to cross the cell membrane. Okay? There are two ways for you to be transported to cross the membrane. Okay? The first way is called passive transport. And passive means without energy. Passively. Right? Like this club again, if it is full, when you open the door, does he make an effort to get out? Just open the door and he'll get out. It's full, it's packed, right? Just open the door, he'll get out. He doesn't need energy, right? But what if you try to get in? Do you need energy? Of course, it's full, it's packed. You have to squeeze yourself in, right? So we call that the active transport. So the passive transport is no need to energy. The active transport is you do need energy. When is it passive? When is it active? Concentration gradients. If you're moving via concentration gradients with the concentration gradient, this is passive, right? Just open the door, passively they will get out. This is passive. So if you're moving from high to low concentration, is that passive or active? Passive. From high to low. 100 person here, nobody here, or one. This is passive. If you're moving from low concentration, like 2 people, to high concentration, like packed club, is that active or passive? Active. Okay? So what determine active or passive? Concentration gradient. Are we following? Did you get this? Passive transport, the first type of transport, which is passive. Does passive need energy or not?
No.
Passive? No. Are you going with the concentration gradient or against? With. Otherwise it won't be passive, right? So we're moving via concentration gradient. Something like this. You look at this green arrow. You have something that have high concentration here. Example, oxygen. You have a lot of oxygen outside and less oxygen inside of your cells. Oxygen will move in your cells, right? Passively, isn't it? Passively. And we call that diffusion. If you're moving via concentration gradient passively, I call that diffusion. A special type of diffusion is diffusion passive or active? Passive. You diffuse from high to low. Is that passive or active? Passive. A special type of this diffusion that's passive is a unique thing for water, and that's called osmosis. So osmosis is the diffusion of water, and the water is going to move like this. Let's say here are two components, and I put a semi-permeable membrane, just like the cell membrane, okay? And I have those two components, okay? I have a liter here, and I have a liter of water here, okay? Let's say I'm going to put a quarter of this salt and half of this salt. Which one have more water? One or two? One. So the water will move like this, okay? What's actually helping it to move is not really because of the water is more here. It is this that's more concentration. Let's say this is salt or sugar, whatever it is. This is what's actually absorbing the water to its side. So you can see it both ways. The water moves from high concentration to low concentration of what? Water. Or low concentration to high in a solute. What's a solute? I talked about this before. If I have sugar and I dissolve it in the water, what do you call the sugar? Solute. What do you call the water? Solvent. And what do you call it together when you mix it? Solution. This is a solution. The salt is the solute. The water is the solvent, isn't it? So which one is more concentrated? The one that's half salt or quarter salt? Half salt. So the half salt is going to absorb. So we can call it both ways. But when you go more like in physiology, you will see that we're referring more to high concentrated versus low concentrated. I have less salt here and more salt here. The more salt will pull the water in to its side. But this is unique for water. You cannot call osmosis for anything else. Anything else that moves from high to low, I can only call it diffusion. Osmosis is a special diffusion for water only. Is that clear? Next type is called facilitated diffusion. What's facilitated diffusion? It's diffusion anyway. So when you say diffusion, it's from high to low, isn't it? Rule of the thumb, you diffuse from high to low concentration, right? So this is diffusion anyway, because you're moving from high concentration to low concentration. Why don't you just call it diffusion if you're moving from high to low? Because this is another special type that the molecules are not small enough to just diffuse in. It's too big, okay? So now it cannot cross. You cannot just diffuse, okay? So what are you going to do now? You're going to facilitate this diffusion using a transport, okay? Like, here is an example, water. I have a lot of water outside, I have a lot of water inside. So the water is going to diffuse in, right? Can it actually diffuse in through the lipid? No, I have to facilitate its entrance. Even though it's high outside and more inside? Yes, it's because of the nature of the water. Did you get my idea? This is a different scenario. Not just water, anything that's hydrophilic. If you're trying to cross, even if it is from high to low concentration, it's called diffusion anyway. It's passive anyway, I understand that. But can it cross, just like oxygen and carbon dioxide? No. This is hydrophilic. It cannot cross regular diffusion. It's not simple diffusion. This is called facilitated diffusion, even though it's going from high to low, even though it's passive, even though I still call it diffusion, but this is a facilitated diffusion. You have to have a protein that facilitate its entrance in. If you try to get it in through the regular parts of the wall, it will not be able to get in. Did you understand this? If you have any question as we go, just let me know. Right? Last type is active transport. What do you think active transport is?
You need energy.
You need energy, and you're moving from
Low to high.
low to high. This is called the active transport. If you're moving against the concentration gradient. So this is called the active, and this is the only one. So the question will come, why are you even trying to move something from low concentration to high concentration? I need more. The cells need more. Like, what if you have a lot of, you have some potassium outside and a lot of potassium inside? Why are you even getting the potassium? The cells need it. And apply that on anything. Like you have calcium, but you need more. Isn't it moving against concentration gradient? Why are you spending energy? Because I need it. Right? To that extent, to cross against concentration gradient and use your energy? Yes. If I need it, I just have to do it. Right? And this can apply in different things, but I'm not discussing the details now, but just giving you examples. If sodium is more inside than outside, but you need the sodium inside for some reason, you can still cross it. Across against the concentration gradient? Yes, I'm going to spend some energy, but I have to do it. Okay? Last type. What if you have something that's too big, no matter what, it will not get in? Something that's big, like it cannot cross the wall, which is a simple diffusion, not even facilitated. You cannot even fit it in that protein, and it's not even active. This is not the issue. The issue is too big. Too big. Like if I wanted to push somebody like who have a big body, and you can push him a little bit and he can fit into the door. That's fine, but can you fit in, or can you get a truck inside this classroom? Of course not. What's the only way to get a truck inside here? You have to break down the wall, right? We call that cytosis, exo or endocytosis. If you're entering a truck into the classroom, you're entering it in endocytosis. Okay? You will break down part of the wall. If you need it, you will do it. Okay? And I call that endocytosis. What if I have a truck in here and I wanted to get it out, or not truck, something that's big enough that cannot go through anything? I have to break down the wall. I call that exocytosis. Okay?
Aren't you destroying the cell by doing that?
You're not destroying, you're just taking part of the wall and then you're going to replace it. So you're not really destroying. Okay? Like this. If I want to fit something in here and it doesn't fit in, can I remove these temporarily and then put it back?
Yeah.
Yes. So this is, you're not literally breaking down the whole wall. You're just making a bigger opening. So you're taking part of the membrane and you travel in. You're taking part of the membrane. Like you go to the membrane is big. You take part of it and get inside. And then we're going to fix it after you get in. Endocytosis versus exocytosis. Look at this. It goes like this, for example. These particles are getting in. This is part of the membrane, right? You're pushing into the membrane and it will take it and leave out. So you have a cut here, right? You have an opening here. We're going to fix it. Okay? Or after you're done, this part can go back and fix it. Okay? I can remove the door temporarily, completely, even the surrounding, and then put it back. Right? So this is endocytosis. Exocytosis is just the opposite. I'm taking this, I want to take this out. No, you can't. It's too big. Okay, I'm going to remove parts or fuse it with the membrane and then take it out. And we call that endocytosis and exocytosis. One more thing to learn here, that when you push something in and they take the membrane with it, you call that a vesicle. Okay? The next part will be the chromosomes, and I think we can stop here. for this part, and we will do the other part next time. How many here? Yeah, we have 8. Okay, so 4 is good enough, and we will do from 5 to 8 next time.